Hello guys, my name is Shmail. Today I'll be going over viruses. So, let's begin. So, viruses. As you may or may not know, viruses are not a cell, so they are known as acellular. And simply they are nucleic acids with proteins surrounding them. So in the exam, whatever you do not put that viruses are a type of cell. If you want to put something like that, just say that they are acellular. And so, if they're not a cell, therefore they're not living. So, an example of these type of viruses are HIV, influenza, and rhinoviruses. So, all viruses reproduce inside the cells of other organisms. So, they don't go through binary fission, they don't go through mitosis, they do not go through meiosis. They go through the reproduction of inside cells, so they pretty much duplicate themselves. So, uh, so what they do is they reproduce inside the cells of other organisms, and these cells are known as host cells. So when viruses attach onto another cell, the cell that it attaches onto is known as the host cell. So once inside their target cell, the host cell, their genetic code is ejected. So what happens is the virus grabs onto the cell and injects its DNA, genetic code. And so the code instructs the creation of more viruses. So during the time it takes the invaded cell to recognize the foreign DNA and destructive activity, as the viruses spread and use up the host organism's resource for tearing the cell apart, the virus has replicated and can spread further whether its host has survived the invasion or not. So basically what happens is the virus injects the DNA and the cell starts to try and figure out what's going on, which foreign activity has taken place. But by the time that's happened, the DNA, in the cell's mechanism will have learned to reproduce the virus, and so multiple viruses would have been produced, and whether the host cell has survived or not, the cell will start to burst, and the virus will spread to other cells as well, and this will carry on, and it's a growing process, so it has exponential growth. So the genetic material, whether DNA or RNA. So let's look at some of the features here. So a capsid, which is pretty much a protein coat, which contains the genetic material. Attachment proteins, so these things here, which direct its invasion of the host cell. So in bacteriophages, so this is what a bacteriophage looks like. It looks pretty much like a spider. And so in the bacteriophage, these are the tail fibers at the bottom, resembling spider legs, so these here. And while in influenza is the ball-like protrusions, so protruding stakes covering the membrane envelope, and these act as antigens that a host organism may have antibodies for, or develop the antibodies and thus immunity as a result of the invasion. So yeah, so you need to know about the protein code, the DNA strand inside, and the fibers, so the attachment proteins, so pretty much the protein code and the proteins on the outside. So let's look at viral replication. So since viruses are not alive, they're acellular, they do not undergo cell division. So instead they inject their DNA or RNA into a host cell. So then the target cell uses its own machinery to replicate the viruses. So in order to inject the DNA or RNA, viruses must attach to the host cell surface. So they can't do anything without attaching to the cell. And to do this, they use their attachment proteins, it's in the name, attachment proteins, to bind to complementary receptor proteins on the host cell. So on the host cell, they also have receptor proteins which attach to attachment proteins. So as a result, some viruses can only infect one type of cell, while others can infect a lot of different other cells. So let's have a closer look here. So you have this circular virus here, which is influenza, and becomes attached to a target cell on an epithelial cell. So the virus attaches itself to the outer membrane of the cell. And so sooner or later the cell engulfs the virus by endocytosis. So you do need to know that word. And viral contents are released, and so the DNA strands are released, and RNA enters the nucleus, where this is the DNA genetic information is duplicated. So then new viral particles are made and released into the extracellular fluid, so outside the cell. And the cell, which is not killed in the process, continues to make new viruses. And so viral messenger RNA is used to make viral proteins. So let's have another look at a simpler diagram. So you have this bacteriophage here, it's a type of virus, attaches itself to the surface using its attachment proteins, and so it penetrates and viral DNA is injected into the cell. The coat of the virus remains outside, and replication nucleic acid is replicated in host cells. So the genetic material is injected, and so some of the RNA goes inside the nucleus and is replicated, and so then synthesis new protein codes are synthesized within the host cell. So protein codes are made inside the cell, and to assemble these, so sooner or later the genetic material will be assembled, the proteins code will be assembled, and soon releases the viruses by cell ruptures. So yes, that's it for today. So I thought you might find that helpful, and if you did, please do comment down below. And so in the next video we'll look at microscopy.